Today we will deal about the gametogenesis in case of the animals or in general what we can say. So this is the class overall 61 and the veterinary anatomy, physiology and biochemistry class 8 for the Kpsc examination for the veterinary officers of Government of Karnataka. So, what is the gametogenesis? That's the first question. And it's a process by which the diploid or haploid cell undergo cell division and differentiation to form mature haploid cells. And that's called as the gametogenesis. So, gametogenesis occurs by meiotic division of the cell by diploid gametogenesis into various gametes or by mitotic division of haploid gametogenesis cells. So, this is further showing the existence of a multicellular haploid phase in life cycle between meiosis gametogenesis is also referred to as alternation of the generations. So it is a process of gametes formation. It occurs in the gonads, ovary, and also in case of the testis. So that's the gametogenesis is divided into four phases. The extragonadal origin of primordial germ cells. Second one is the proliferation of germ cells by mitosis. Third one, meiosis. Fourth one is the structural and functional uh, maturation of the ova and also the spermatozoa. So the gonadal cells are the specialized cells and they enter the meiosis and replicate by mitotically, genetically identical to the original zygote. So when they begin meiosis, they are called as the spermatocytes and oocytes, depending upon the male and female respectively. So gametogenesis in case of males it is called as spermatogenesis, oogenesis in case of the females. So spermatogenesis is a process in which the formation of male germ cells called as the sperm formation. It occurs in the seminiferous tubules of the testes and it is thread-like in case of shape. So this is the process in which the gametogenesis of spermatozoa takes place. So this is the lumen of the seminiferous tubule outside, and all these are spermatozoa, and these are called as the Sertoli cells, which produce the spermatozoa, and the structures which are not yet formed, the fully mature spermatozoa are called as the spermatids, and the other things are called as these secondary spermatocytes, and the spermatogonium is the main cell which gives generation to primary spermatocytes in which a tight junction is present and which gives the secondary spermatocyte generation. Then spermatids are further formed, and these are all called as the Sertoli cells. This happens especially in case of male gonadal organ, that's also called as the testes. Then spermatogenesis occurs from puberty to old age, producing immense number of spermatozoa at an average of 1.5 million spermatozoa per minute. It's a huge number. So the average rate of spermatozoa production is 1.5 million or 15 lakhs spermatozoa per minute. The sperm will mature in epididymis, nourished by Sertoli cells for up to 10 weeks. They will be there in the epididymis. Humans aged about 13 to 90 can make 1 billion sperms per a day. So every human being aged about 13 to 9 can make 1 billion sperms per day. It's a great number. The serotonin cells are supporting cells that have several functions in the genesis of the spermatozoa, they form the blood testis barrier, nutrients and circulating substance, not to directly to reach the germ cells. The Sertoli cells determine which substance reach the germ cells, whereas the spermatogonia are outside the blood testis barrier. And here you can see the spermatogonium or spermatogonia are outside the blood testis barrier. Then the they form the blood testis barrier, nutrients, etc. Then they also produce antigen binding protein, which are necessary for spermiogenesis. That's the morphological development of spermatids to spermatozoa. So this is the one spermatogonia. They are the first cells to give birth to secondary spermatocytes, and there are two numbers. So here also the primary spermatocytes are divided. So the first meiotic division takes place and secondary spermatocytes are produced, and second meiotic division, then spermatids are followed, and finally the spermatozoa are produced, or the mature spermatozoa are produced, and they migrate to epidermal cells and grow there ultimately. Spermatogenesis is the process by which the male primary sperm cells undergo meiosis and produce a number of cells named as spermatogonia, from which the primary spermatocytes are derived. Each primary spermatocyte divided into two secondary spermatocytes, and each secondary spermatocyte into two spermatids or young spermatozoa. And these develop into mature spermatozoa, also known as the sperm cells. Finally, the primary spermatocytes give rise to two cells, the secondary spermatocytes and two secondary spermatocytes by their subdivision to produce four spermatozoa, as we have seen in the earlier slides. Then spermatogonium is the intermediary male gametogonium, that is the first cell, a kind of germ cells or germ plasm, diploid in number 44 autosomes, and two sex cells will be present. So diploid in two numbers. It is very small and under the process of development, undergoing the development. So primary spermatocytes diploid in number 44 autosomes. So they are also undergoing increasing the size, undergo G2 phase. Secondary spermatocytes haploid in number 22 autosomes, double standard, and one sex cell each. They can see the cleavage of furrow undergoing the meiosis, one that's called as the reduction division, and these chromosomes are reduced into half number. So the spermatids are haploid in number, and 22 single-stranded autosomes and one sex cell will be present and undergoing the meiosis second or equational division. So finally spermatozoa are produced. So it is a motile sperm cell or moving from a form of the haploid cell, that is the male gamete. It joins an ovum to form a zygote, and mature sperm cells are ready to fertilize the egg or ovum. And oogenesis is the one which happens in the female one. So oogenesis is the process of formation of the female germ cells or egg formation. It happens in the ovary or the female gonadal organ. It is spherical in case of shape, and oogenesis begins in early fetal life itself. So, very early fetal life itself, all oocytes formed in females are produced during the fetal life itself, and many of them degenerate with time. And at birth, the ovaries contain about 2 million oocytes, 2 million oocytes, and all oocytes go into meiotic arrest when they reach the first meiotic division during the fetal life. So the primary oocytes remain in the prophase of the first meiotic division until the time of the puberty, when they are gradually released to complete meiosis at regular intervals known as ovarian cycle. So usually the average only one oocyte matures during each cycle, which occurs approximately monthly intervals, so that the amount of the oocytes to be ovulated is about 50,000. No, what is this oocytes? Uh, 5,000 oocytes in the life span. So here you can see oogenesis is by oogonium, just like this spermatogonium, then ovary, then it is going to many oogonia are produced. Then before birth, this process will be there. Then after birth, the ovary is going to have the different ovulation process. So diploid number 44 autosomes and two sex cells. So it is very small and under the process of development and undergoing the GS phase. Primary oocytes. So it's a well whose ovary function is to divide by the process of meiosis. So it's also diploid in number and 44 autosomes and two sex cells, increasing the size undergoing G2 phase. So secondary oocytes, so they also undergo meiosis 1 and chromosomes are reduced into half number. And always the begins during the embryonic development but halts the diplotin stage of the prophase 1 until puberty. And primary oocytes that continue to develop in each menstrual cycle. However, synapsis occurs and tetrads form, enabling chromosomal crossover to occur. So, results of meiosis, the primary oocyte has now developed into the secondary oocyte and first polar body. So, oocyte or ovum, immediately after meiosis, the haploid secondary oocytes initiate to equation division, and this process is also halted at the metaphase 2 or stage 2 until fertilization, and such should ever occur. And when meiosis 2 has completed, an oocyte and another polar body have been created. So the polar bodies, both polar bodies disintegrate at the end of the meiosis 2, leaving only the oocyte, which when eventually undergoes the mature ovum. So this is the function of the polar bodies to discard the extra haploid cells of the chromosomes that have been resulted as a consequence of meiosis. So there is a comparison here. The spermatogenesis both are also initiated from the germ cells called as the spermatogonium and undergo different cell division and ultimately either X or Y spermatozoa are formed, whereas in case of the oogenesis only X is formed. So that's about the present topic, and thanks a lot for watching the particular lecture and study well. Have a nice day. Bye.